Okay. So, yeah, good evening, everyone. Today, we will be talking about the week 3 lectures. So, those who are new to this class, I am Srijan Sen from IIT Kharagpur, and I will be taking your week 3 live session on the course Fundamentals of Protein Chemistry that is being taught by Professor Shagota Dasgupta of IIT Kharagpur. So, I'm sharing my slides with you all. So I guess my screen is visible to you all, and like the way I usually teach, it's first I go with the brief introduction to the lecture slides, and then I discuss the question and answers. Okay. So let us start with our class today. So in the first 2 weeks, what we actually discussed was the basics of proteins and mostly amino acids, the different types of amino acids, the protein structures, the different structures of proteins and all. So we will be talking today about mostly the protein structure, stability and denaturation of proteins. Right, so we are going into a little bit of the depth of proteins because we need to understand we had talked about the structure, but also we need to understand about the stability, denaturation, renaturation of proteins, and mostly about the different types of proteins that are actually available or very commonly known to us. So we have seen this in the last class where we discussed about the primary sequence of proteins, where we have, so the primary structure is basically composed of the different amino acids joined by polypeptide chain, the peptide bonds. Then comes the secondary structure where we have the local folding of the residues, giving us mostly the alpha helices, beta strands, the beta turns, loops, etc. Third is the tertiary structure where we talked about the domains and the motifs, like the two or more secondary structures coming together to give us a tertiary structure. That's the long range of folding. And then when we get a three-dimensional structure, so multiple three-dimensional structures combined together to give us a quaternary structure. So mostly the quaternary structures are because of the functionality, where the proteins that are in the monomer form are usually not very active. Rather, the quaternary structure or a multimeric structure organization is absolutely needed for the carrying out of different functions, like we discussed about hemoglobin. Okay. And lastly is the supramolecular structure. So these are large scale assemblies for different functions and all. Right, so structure determines function, we all know that. So what determines the structure? So the energy and the kinetimatics is controls the protein folding problem. So how can we determine the structure of proteins, and why are these proteins very important? Because proteins are important in carrying out functions like cellular regulation, cell signaling, the maintenance of the structure of other proteins. Then there are some certain signaling molecules like transport proteins, then movement, catalysis, enzyme catalysis. So enzymes are also proteins. So for enzyme catalysis, we require the structure needs to be certainly determined, because the structure determines the function of a particular protein. Now, how can we determine the structure? To determine the structure, there are several experimental methods, we all know that, as well as there are computational predictions. So among the experimental methods, we have NMR crystallography, NMR spectroscopy, X-ray crystallography. Then nowadays, it's very trending nowadays about the cryo-EM microscopy. Okay. And apart from that, about the secondary structures and all, you can get an idea through CD spectroscopy. And there are several many experiments which you can actually do to get an idea about the structure of a particular protein. Even a part of the structural protein can be of an ordered protein can also be achieved. Now, the computational prediction comes from via through algorithms. Now, these algorithms are formed by different labs, different research groups and all. So one such example you all might be knowing is the AlphaFold. So AlphaFold gives a very accurate structure of different proteins, of mostly ordered proteins. However, it fails to give structures of proteins that are mostly intrinsically disordered or are very flexible to a certain point, because it's very difficult to know the structures of these proteins, because they are dynamically so flexible that the understanding the structure of these proteins is a really big challenge. Okay, so these are the different ways of how you can get an idea about the structure of a particular protein. What we are more interested in today is about the protein folding. Okay, So the amino acid sequence of a protein determines the 3D fold. So this was said by Alphinson in the year of 1950. However, there are some exceptions that all proteins can be denatured. Some proteins can have multiple conformations, and some proteins gets folding with the help of chaperones. And the function of the protein is determined by its three-dimensional fold, or the folding of the protein determines the function of a particular protein. Now, can we predict the 3D fold of a protein given its amino acid sequence? We definitely can. Of the development in the fields, in the research field that has happened, we definitely can predict the three-dimensional structure of a protein via computationally, by carrying out different algorithms. There are different systems that can, just by inputting the amino acid sequence of the particular protein, you can get back the structure of that particular protein. So it is definitely possible to get the 3D structure. And AlphaFold, so previously there was just the AlphaFold. Now there has been different updates in that system, and from there AlphaFold 3 has been generated, and you can easily understand the structure of the protein through just by inputting the amino acid sequence. You get even the accurate of these folds, right? Now, what is the protein fold? How do you understand this protein folding, or what's the concept of this Leventhal paradox? So, in the last class, I said a little bit about this Leventhal paradox. So it's similar to, so I'm telling once again. So, like for example, if you have a protein of 100 amino acids, and each of these bonds, or each of these residues can actually assume three different conformations. So it's 3 to the power 100. So there can be around 10 to the power 47 conformations that can be attained by one particular protein. And if you consider that these change in conformations happens in the fastest of the motions, that is 10 to 15 seconds, it would take around 10 to the power 32 seconds to actually complete an entire of the conformation and get to a particular structure of a protein. So if this is the real scenario, that it takes 10 to the power 32 seconds, it is equivalent to 10 to the power 25 years. So if we are assuming a protein as big as 100 amino acids of having three different conformations, then it is supposed to take 10 to the power 25 years, and that's a huge time. Probably in our lifetime we would never be able to see the folding of a protein, but that does not occur. So each protein folds quickly into a single stable conformation, and that is the stable conformation that it attains. So once it gets to the stable conformation, it sticks over there, and from there the 10 to the power 25, that huge number, is reduced to probably another few seconds. And so the protein folding takes a very less amount of time, and maybe in a few seconds you get the protein folding, and that is where the concept of the Leventhal paradox comes. So the different energies or interactions that are present in stabilizing the protein folding includes the hydrogen bonds between the neutral groups. Between the peptide bonds, there are hydrogen bonds. Between the secondary structures, there are few hydrogen bonds between alpha helices, beta strands. We all know about that. We had seen that in the last class. Then there are a few ionic interactions, charge-charge interactions, electrostatic interactions. There can be attraction, repulsion, hydrophobic interactions that control the core of a particular protein. And these interactions have different strength in kilojoules per kilocalories per mole. Like you see, the hydrogen bonds have strength around 5 kilocalories per mole, ionic bonds around 10, hydrophobic interactions are 2 kilocalories per mole, van der Waals interaction 1 kilocalories per mole, and then you have the covalent bonds like the disulfide bonds that are 51 kilocalories per mole. Right? So these interactions are bondings. These bondings or interactions are the ones that control the folding of a protein, that control the stability of a protein, and maintaining the structure of a protein, because structure determines the function. We are all concerned about the function of a particular protein. The structure may be whatsoever, as long as the protein is functional, we are happy with it. So what is the assisted folding of proteins say? So not all proteins fold spontaneously as they are synthesized in the cells. Folding of many proteins is facilitated by a specialized class of proteins called the chaperones. So chaperones are heat shock proteins, so HSP70 type chaperones. Then you have the DNA K, DNA J. So these are the different chaperones that are play very important role in the folding of proteins. So that is the class 1 proteins, where we have the heat shock proteins in the cells. The second one includes your chaperonins like the Groels, Groes. So these are the another class of chaperones that also help in the folding of a particular protein. So the major function of a chaperone is to assist the proteins to attain a particular structure through folding. Now there are also other enzymes that are involved in protein folding, like the protein disulfide isomerase PDIs, protein prolyl cis trans isomerase, the PPIs, and these control the protein folding spontaneously. Now, the conformation of a native protein is marginally stable. There are several different conditions in the cell that can cause the protein structural changes as well as loss in the structure. The loss in the structure of a protein is what we know as the protein denaturation. So denaturation means the protein is opening up. The energy that controls the interactions that hold the protein in its folded state, in its native state, is basically lost, and the protein is completely opens up and is said to be denatured. So in the denatured state, the conformation of a protein need not be completely randomized. It can be folded partially unfolded, and that can happen. So what are the conditions that control this protein denaturation or play an important role in the protein denaturation? So a number of physical and chemical reagents can cause protein denaturation. So one such agent is the heat. So heat is one of the biggest or the classical agent. So when you provide heat to a protein, it basically opens up because it the bondings because in when the temperature increases, the bonds break, the interactions breaks, and that leads to opening up or unfolding of the protein, leading to protein denaturation. So the classic agent like heat, which has complex effects on many weak interactions in a protein, primarily the hydrogen bonds. Now on heating, a protein's conformation generally remains intact until an abrupt loss of structure occurs over a relatively narrow temperature range. So you see over here, so these are the graphs of two different proteins. One is ribonuclease A and the other one is apomyoglobin. So what is this Tm? So Tm is the melting temperature. So the temperature at which a protein is 50% folded and 50% unfolded, that is its melting temperature. So when it is 50% unfolded, that is its Tm. So for like you can see for a ribonuclease A, the Tm is around 56, 57 degrees. Whereas for apomyoglobin, the Tm is around 62, 63 degrees. Right? So the abruptness of the loss of structure suggests a cooperative process in which loss of structure in one or more parts of the protein rapidly destabilizes, and that destabilization leads to the unfolding of the other portions of the or other parts of the protein as well. Now, there are certain chaotropic agents as well as in the changes now extremes of pH, then miscible organic solvents like alcohol, acetone, certain solutes such as urea, guanidine hydrochloride, detergents, these all can affect the folding of a protein, and the protein basically reacts to these certain changes, and it basically denatures. Now, organic solvents like urea and detergents act by disrupting the hydrophobic interactions that are important. So, hydrophobic interactions are important for forming the core of the particular protein, and that is why And urea and detergents, what they do, they affect these hydrophobic interactions that are important for holding the core of the protein, and it causes a nuisance over there, and that is why the protein basically gets unfolded. Now, the extremes of pH alter the net charge on the protein, causing electrostatic repulsion and the disruption of some hydrogen bonding. And when some certain weak interactions gets disrupted, it leads to the unfolding of the protein. Now, the denatured structures resulting from these various treatments are not necessarily the same, and denaturation often results in aggregation and precipitation of the unfolded protein, because you see that what happens is, like for example, you are increasing the pH or decreasing the pH. What happens, the protein has reached to its pi, and that is the temperature at which, or that is the pH at which the protein basically aggregates and unfolds, right? So these things can basically occur, right? And apart from that, whenever you are treating with some chemotropic agents like urea or guanidine hydrochloride, these are very soluble, so these are basically used for these are either reducing agents, or they are important for solubilizing your protein. So how does your protein get solubilized? It just entirely opens up the protein, and that's how your protein gets solubilized. Now, this is about the denaturation of a protein. So that happens. Now, how much treatment, what is the concentration of guanidium that you are using, or what is the concentration of urea that you're using, at which the protein is unfolded, right? So how do you know how much guanidium is used? So probably you start increasing the guanidium concentration, and you plot a graph. The point at which the protein is 50% unfolded, that is the point, that is the middle point. So at that particular guanidium, or at that particular amount of urea, your protein is basically unfolded, which you can see from the lower graph. Okay. Now, denaturation is one part of the story. We need to get the protein renatured as well, because that is the state in which we require our protein to be. So early studies where the renaturation of an enzyme ribonuclease was done, it was determined by its primary structure. So you see over here, this is the native state of ribonuclease A. These 58, 110 are the different cysteine residues through which the disulfide bonds were formed. An addition of urea and mercaptoethanol entirely unfolded the protein. So the unfolded state is inactive. In the presence of the reducing agent, the disulfide crosslinks cannot be formed due to the reduced cysteine residues. Now, upon removal of urea and mercaptoethanol, you get back to the native chemical structure. Now, in this experiment, what was done is, like, the purified ribonuclease was denatured to its unfolded state by the exposure of concentrated solution of urea and the reducing agent mercaptoethanol. The unfolding of the protein, uh, so this unfolded the protein and reduced all the crosslinks to the cysteine residues. Now, when urea and mercaptoethanol were removed, the randomly coiled denatured ribonuclease spontaneously refolded to its catalytically active form. Even the proteins for a unique disulfide could be formed, like it has 8 cysteine residues, so there can be combinations of 4 disulfide bonds, but yet it when you are trying to actually push the folding of a protein, now when folding of the protein is very fast, it forms random disulfide bonds that can lead to the disruption of structure. But when you're giving time for the protein to fold, it gets an information about the proper folding of the protein, because one bond forms, the other does not form, and it again breaks, and again reforming of the disulfide occurs. This process happens as long as the correct conformation is not attained. So when the correct conformation is attained, that is the conformation in which all the molecules of the protein try to go. So folding is actually a slow process. You need to give time to the protein to fold. You cannot be like quickly you want it. Once you give time to the protein to correctly refold, you get back the native catalytically active state of that particular protein, and that was actually seen in the previous experiments. So what is the thermodynamics behind the protein folding? So the protein folding process can be viewed as a kind of an free energy funnel. So the unfolded states are occupied by the top of the funnel, and these are characterized by high degree of conformational entropy and relatively high free energy, because the proteins are in a very much uh, entropically very high and flexible enough, because they are in an unfolded state. Their mobility is very much high. As folding proceeds, the narrowing of the funnel reflects the decrease in the conformational space that must be searched as the protein approaches its native state. So small depressions that you see over here along the sides of the free energy funnel, these are the semi-stable intermediates that can slow the folding process. That is what is required to get to this narrowest of the range, because you need the protein to be absolutely folded. So there are few semi-stable states. So the funnels can have variety of shapes depending on the complexity of the folding pathway, the existence of semi-stable intermediates, and the potential for particular intermediates to assemble into aggregates of the misfolded protein. So ultimately your goal is to reach at the bottom of this funnel, because this is the most stable state of the protein, but in the middle there are multiple ups and downs. So these are the semi-stable intermediates. So you see over here, this is the beginning where the entropy is very very high, and then as the energy decreases, so the energy decreases as the protein starts folding. So the percentage of the residues in the native conformation, so most of the residues when they get into the native conformation, you start the uh, funnel starts getting narrower, and ultimately when there is a proper discrete folding, some intermediates can be there, here and there, because after all there are a few flexible loop regions, flexible residues that are here and there. So because of that you see a few bumps here and there, but ultimately the native structure gets formed as the energy decreases, the entropy gets lowered, and most of the residues are in their native conformation, and this is the native structure of the particular protein. So, as we were discussing about chaperones, so many proteins do not fold spontaneously as they are synthesized by cells. So, the folding of these proteins requires the general class of proteins called chaperones. Chaperones interact with partially folded or improperly folded polypeptides, facilitating correct folding pathways or providing microenvironments in which folding can occur. Now, several types of chaperones are found in organisms ranging from bacteria to humans. The major classes are, like I was saying, the heat shock proteins. You have HSP70 family of proteins, chaperonins, like the Groell's HSP60 family of proteins. And these proteins are induced in bacterial cells by exposure to high temperatures. And molecular chaperones act as monomers, where chaperonins are large multi-subunit complexes. So these chaperones are itself some protein molecules. Now some act as monomers, the others are multi-subunit complexes, the quaternary structures. Like I said previously, some proteins do not act like some proteins cannot act like as a single-handedly as a monomer. They require multi-dimensional structures, multimeric structures. So that is one such example is the chaperonins. Now for structure prediction, you have experimentally you can go through X-ray crystallography, NMR spectrometry, then cryo-EMs. All of these can be used to do experimentally. Computationally, there are so many systems nowadays, like I was mentioning about AlphaFold. These sequences implies the structure, therefore in principle we can predict the structure just from the sequence alone, and we can just by looking at even the sequence of the protein, we can there are some tools, bioinformatic tools, that also gives us about the energy, geometry, kinetometrics, about the physical characteristics of the particular sequence, about the particular protein. Okay, and then based on homology we can find the best match in the database, because some proteins belong to a particular family, belong to a particular class, and depending on the homology of that particular of the known protein, we can get an idea about our protein of interest. Mm, Now there are a few tools like Rosetta. So new structures generated by swapping the compatible fragments. These are computational tools. So the accepted structures are clustered based on energy and the structural size. The best cluster is one with the greatest number of conformations within 4 angstrom RMSD. So what is RMSD? It's the root mean square deviation. So for example, you have an atom present in column 1 and the another atom goes in column 2. So it's like the deviation that is occurring between the columns, so between the atoms. So it calculates the deviation of the atoms. It calculates that, and it does it for every atom, and you get it as a combination of the entire protein. So the lower the RMSD value, it's good for your protein, because that means your protein is less flexible, as well as it is present in its native conformation. It's present more or less in its native conformation. So the best cluster is the one with as low as 4 angstrom of RMSD deviation. Some proteins are so stable that even if you get less than 1 angstrom deviation, and that's a fantastic thing. So you do some molecular dynamics. So these are the understanding of the dynamics of the protein. You do some molecular dynamics to get an idea about the root mean square deviation, root mean fluctuations of the different residues. So there are multiple analysis tools. Like the deviation gives you an idea about how much your protein is deviating at the different time points. Like in the quadrant 1 comma 1, your protein is there, then it goes into 1 comma 2, so there is a good amount of deviation. But if it does not deviate much, you see the number is going down. And then you have the root mean square fluctuation, where you can understand, like, RMSD is for the entire of the protein. Now if you want to know which residue is fluctuating a lot, you can do RMSF. That will give you a residue wise fluctuations. Right, so there are different tools through which you can get an idea. Now, homology-based prediction tells you, so you align the query sequence with the sequences of the known structures, usually greater than 30% similarity. You superimpose the aligned sequence onto the structure template, and according to the computed sequence alignment, you get the sequence alignment, you get the superimposition, and from there you can understand how much your protein has deviated, or what are the secondary structures that your protein might be having which is not present. And even if it is having, it is fantastic, because then you know that your protein is present in its native structure. Also, we can perform local refinement of the resulting structures in 3 dimensions. So the number of unique structural folds is small, possibly a few thousand. 90% of new structures submitted to PDB in the past 3 years and have similar folds in PDB. So like I was telling, so homology based prediction, just for that you need to know such a protein that belongs to the family of your protein of interest, because only then the folds will be the same. And once when you get an idea about which stretch of amino acids is going to give you an alpha helix, or which stretch is going to form a beta strand, that is very important to know, because only then you can figure out whether it matches your known structure or is it deviating from your known structures, and from there you can get an idea about the structure. But now it's very homology based prediction is a very old age time thing. So once if you know that your protein, so just you can give the sequence input in the alpha fold, and it just gives you back the structure in a few minutes. Okay, and it's very easy, because even if your protein is unfolded, it can give you a random structure, but the folding or the folds that are present in the protein is actually maintained, because after all it studies the previously known structures, as well as it studies the sequence in great depth, and the algorithm is actually formed in that manner, where you can get the folds of of a very folded protein to a great accuracy. Okay. So yes, that is all from the class. If you have any questions, you can ask me. After this, I will be going to the question and answers. If you have any doubts, go ahead. Which slide are you? Sorry, missed the text. Which slide do you want me to repeat?
Actually, this slide, after discussing about comparison of bond stance, now it is far behind it.
Which one?
This one.
The Leventhal paradox?
Yes.
Okay. So the Leventhal paradox, what does it say? Like, for example, if you have a protein of 100 amino acids and it can attain around 3 different structures or different conformations, let us assume that. So upon taking such an assumption, it's like 3 to the power 100, because each residue can undergo 3 different conformations, and if we consider that there can be 10 to the power 47 conformations that a protein of 100 amino acids can actually attain. Now, if we are assuming, because this is entirely based on an assumption, like each conformation takes around 10 to 15 seconds for sampling, then it would take around 10 to the power 32 seconds for actually one particular protein to fold. And 10 to the power 32 seconds is equivalent to 10 to the power 25 years. But in reality, 10 to the power 25 years is a big number. It is not possible for us to then even observe the particular folding of a protein, or a particular protein will never fold probably in even 100 years. So a person or a researcher can never do a work with a particular protein if it would have taken such a long time. But this is not the story actually, because this is where the concept of Leventhal paradox comes. Once when a protein reaches a conformation that is more stable than its previous conformation, it sticks to that. Like when you know that this is the best part, so this is better than my previous conformation, I will be in that, and then the entire process carries on. However, this 10 to the power 25 years is a very big number. Like I was showing you this particular graph, this inverted funnel. So once when it reaches this particular steps, you might assume, then it does not go back to the previous conformations, because this is the most stable structure among the all different samplings, all different conformations. So it will try to even stabilize whatever it is, whatever structure it has, it will try to even stabilize it more, not destabilize. So ultimately, this from the top of the funnel to go to its native structure, it takes probably a few seconds, even a few milliseconds. You can assume that. So that is where the Leventhal paradox comes, because 10 to the power 25 is a huge number. It is actually not a real number. But had it been like every conformation is occurring individually and happening, 10 to the power 25 would have been more realistic. But it's not the story. It takes around a few seconds for the folding of a protein. Okay.
Okay, sir. Thank you.
Anything else? Anyone else? I'm not very sure about what advancement has been taken, but what I have noticed when I have used AlphaFold 2 versus AlphaFold 3 is about the prediction of more, because the ordered uh, parameters, the for a folded protein, you see whatever folds are there, it is basically the same in AlphaFold 2 or 3. For a folded protein, it can predict it more accurately. Even in AlphaFold, when AlphaFold first came, it could do that. What I am actually happy about is when it actually does it for the intrinsically disordered proteins. Okay, because previously it was never able to do it for a disordered protein. It used to give us back with an accuracy of zero point 0.5, maybe a 50% or 40%, but the number has actually increased to around 70-80% for the intrinsically disordered proteins. Though it's very difficult to say, because you know why? Because intrinsically disordered proteins themselves do not have a fixed structure. So uh, just for our happiness, it's like AlphaFold 3 has giving a more accurate structure, so we are happy about it. So this is the best confirmation it can have. So it's just the development in the algorithm that they have tried to do to give a more accurate of a structure uh, for proteins that are mostly disordered. But for folded proteins, I do not see a lot of a change. And another thing that I think AlphaFold has also introduced, like previously, AlphaFold used to do it for linear proteins, okay, proteins with free N and C terminus, but AlphaFold 2 AlphaFold 2 or might be AlphaFold 3, I'm not very sure. They have combined with some algorithms, and they have introduced some different algorithms, and they are trying to also develop cyclic proteins and cyclic peptides. Previously, this was not there. So you can, if you want to make some cyclic peptides or cyclic proteins, it is possible to do nowadays using AlphaFold. So yes, it has developed to a certain extent. Okay.
Okay, sir.
See, if you are inputting the structure, so it can also do for membrane proteins. I'm not very much sure about. It varies because it does not consider whether it is a membrane protein or it's an extracellular or an intracellular protein. It just observes the structure, the sequence of the protein, and it goes ahead with the structure. Right. Even if you are giving a membrane protein, probably there will be some disordered regions. There will be some portions that are well folded. So you can get for the folded portions, you will get with a high accuracy. For the portions that are less ordered or a bit more flexible, for that the prediction might be vary. But I would say, as of now, you can go ahead with the AlphaFold. That's the best model that's giving best models according to me. Right. I think I can discuss I can take doubts later on, maybe. Let's go with the question and answers. Okay, like I do, you people give me an answer first, and then maybe I will give you the correct answer. So first you people can try. Okay, and everyone should try to answer, because that will be good and that will be more interactive. Okay, so which of the following residue pairs does not form a salt bridge in proteins? Which of the following residue pairs does not form a salt bridge in proteins? Glutamic acid, leucine; aspartic acid, lysine; aspartic acid, arginine; glutamic acid, lysine. Very easy question. I've got one answer. Okay, this is very easy. So salt bridge interaction occurs between the charged proteins, or differently charged, a positive negative or a positive negative, or a negative positive, something like that. So you see, for all the B, C, D, there is a negative positive, negative positive, and a negative positive. So there is a charge interaction that is occurring. So all B, C, D can form salt bridges. The glutamic acid and leucine cannot form a salt bridge. So option A is the correct one. The next one says a point mutation in hemoglobin causes sickle cell anemia by changing a glutamate residue to a valine. Which type of protein structure is affected by sickle cell anemia following this mutation? So you have to identify which protein structure is getting affected due to a glutamate to valine mutation. Is it the primary, secondary, or tertiary? Or maybe all of the three? So how many people are there? Everyone try to answer. There are not many people, but yes. So if you see that upon introducing yeah, someone raised a hand. Tell me. Okay. Yeah, you can just drop the answer. Right. So whenever you're introducing a mutation, what happens? So the mutation is basically introduced in the primary structure. So the primary structure is definitely affected. Now, if this glutamic residue is involved in a alpha helix or a beta strand, then the secondary structure also gets affected, which is indeed the case in for a hemoglobin. Now, this glutamic residue also gets involved in the secondary structure. That is why introducing a mutation in that position affects the secondary structure. So the primary is affected, the secondary is affected, and ultimately when the secondary structure gets affected, the motif gets disrupted or the domain gets perturbed, and that is the reason where the tertiary structure is also hampered. So 1, 2, and 3 all are affected. Like the primary, secondary, and tertiary, all three will get affected whenever there is a mutation happening. So the option number D is correct over here. The next question, which of the following options contains all the information necessary for to fold a polypeptide chain into a three-dimensional structure? So which of the following options contain all the informations for a protein folding, you can consider for a three-dimensional protein folding? DNA sequence, histone, amino acid sequence, or an RNA sequence? So just by looking at what you can tell about the folding of the protein. Right? Absolutely. So the amino acid sequence, the primary structure of the protein, is absolutely required. It contains all the informations for the understanding the three-dimensional folding of a protein. That is correct. What is obtained after X-ray diffraction of proteins? Molecular density map, nuclear density map, electron density map, atom density map. It's the very basics of X-ray diffraction. So what map do you get from a X-ray diffraction? C or D? Okay, D. You have said D. Okay. So no. So the correct answer is electron density map. So the electron density map, it is a three-dimensional plot that shows the location of space electrons within the repeating units of a crystal. It is basically so when you are actually determining the extra structure, so you get a electron density map where the arrangement of the electrons will actually tell you about the repeating unit of the crystal, and from the repeating units of the crystal you have to draw, you have to analyze and then get the structure. So the from X-ray the end product is basically the electron density map. Looking at the repeating pattern of these electrons arranged in space, you can tell about the structure of the protein. So the electron density map is correct over here. Okay, next one. From the above mentioned curve, melting temperature of the protein will be. Okay, so what will be the melting temperature of the protein from this particular curve? Others please try. Others please give an answer. There is no problem even if you are giving a wrong answer. At least I would be happy that people are trying. Okay, so the melting temperature. So what is the definition of a melting temperature? Melting temperature is that particular temperature where a protein is 50% unfolded and 50% folded. So the temperature at which a protein gets 50% unfolded, that is the melting temperature of a protein. So you see that is the fraction of unfolded protein. So when fraction is 0.5, so at this particular point the temperature is 45. So at temperature 45 degrees, you see the protein is 50% unfolded. So that is the temperature, that is the melting temperature of the protein. So option C. Next one. The molten globule state of a protein is one of the states in the folding process. Which of the options is incorrect about the molten globule state? It is a denatured state, it is a hydrophobic interactions among the nonpolar residues, it's less compact, or it is a collapse state. So which of the option is incorrect? You have to tell me that. So where is this molten globule state basically located? It is mostly in between the denatured and the native state of the protein. Somewhere in between. So which of the option is incorrect? Option A. Yes, that is the incorrect option. Means, yeah, that is the correct option, but that is the option that is incorrect among the three, because molten globule state is that particular state where the protein is not fully folded or not fully denatured. So it is not a completely denatured state. There are several interactions that are going on among the residues. Yes, that is absolutely correct. So it's a less compact and a collapse state, and there are several interactions that are going on to stabilize the particular protein. So therefore, it is not a completely denatured or a completely natured state. So we cannot. So option A is the incorrect option among the three. Now the next one, collagen is found in connective tissues such as tendons, cartilages. We talked about collagen in the last class little bit. Collagen provides the strength due to. So what does the strength of collagen comes from? Alpha helices, presence of EF hand type motif, helix turn helix, or beta sheets. C, A, okay. So what is a collagen? So collagen are coiled coil protein. It's a coiled coil. Why is it a coiled coil? Because the entire of the amino acid sequence is in such a manner like the entire of the three chains. So it has three chains. So all of these are the three alpha helices, and these three alpha helices are intertwined among them. So it's not like three separate alpha helices connected by something or like that. So the three alpha helices are intertwined. So it's a coiled coil protein, and it comes from, so the strength of it comes from the alpha helices. Option A. The next one, in the zinc finger motif, which residues participate in the coordination of the zinc ion? So we are talking here about the zinc finger motif. So which residues are present over here? Yes, collagen has a triple helical structure. That is absolutely right. Any idea about the zinc finger motif, or which residues play an important role? No? Okay. So option A is correct. Cysteines and histidines are mostly present for coordinating with the zinc ion in the zinc finger motif. Okay, so there is nothing much to tell about here because these are just simply two residues that play an active role. Okay, we go to the next one. Protein folding is a spontaneous process. Because why is it protein folding a spontaneous process? Delta G is positive, delta S and delta H are both positive, delta S is negative and delta H is positive, delta S water is positive and delta H is negative. So what is the criteria for a reaction or an entire process to be spontaneous? What is the criteria? So let us go with the options one by one. So we all know delta G equals to delta H minus T delta S. Delta G is positive indicates a non-spontaneous process. Delta G has to be negative. For delta G to be negative, what do we require? We require delta H to be also negative. So delta H being positive is incorrect. Delta S and delta H are both positive, can be partially correct depending on what are the values, right? If delta S is very high positive and delta H is very less positive, it is possible, but as it does not say, so so A, B and C are incorrect. Why is option D correct? Because number one, delta H is negative. Okay, and in protein folding, hydrophobic collapse of the polypeptide chain leads to increase in the entropy of water, and change in enthalpy is large and negative, so that is one thing. And protein folding is mostly driven by enthalpy, so that is why delta H being negative and delta S is positive. So ultimately delta G is negative and it is a spontaneous process. So delta S positive comes from the hydrophobic core. So once the hydrophobic core is formed, water molecules are released from the center, leading to increase in delta S. Okay. Which of the following amino acid sequences is most likely to be a part of the core of the protein? In other words, which combination of amino acids are most hydrophobic? I made it simple for you. So which combination of protein amino acids are highly hydrophobic? So the first one is like lysine, proline, methionine, cysteine, and glycine. The next one is phenylalanine, histidine, methionine, cysteine, and valine. Then you have lysine, proline, serine, threonine, valine, and arginine, histidine, methionine, glycine, and valine. So phenylalanine, histidine, methionine, cysteine, and valine have non-polar and hydrophobic side chains. Right. So we have a combination at number B that will be basically present in the interior of the protein. So option B is correct over here because all of them have non-polar and hydrophobic side chains. The next one, the homology modeling algorithm is applicable for proteins belonging to same family, proteins belonging to different families, proteins with similar amino acid sequence, proteins having same secondary structures with different linkers. Very interesting question. So the homology algorithm will work for which of these? Okay, so proteins belonging to the same family, that is correct. Proteins belonging to different families, that is not correct. Proteins with similar amino acid sequence is also correct. And proteins having same secondary structures with different linkers will also be correct. Because even if you have the similar kind of a fold, you can use that in the homology modeling algorithm. So option C is correct over here. What type of reaction occurs when a disulfide bond forms? Oh, this is an easy question. Which of the following reaction occurs when a disulfide bond forms? So disulfide reaction. So when under what condition can a disulfide bond form? Yeah, absolutely. It's an oxidation reaction that is occurring. So the cysteine gets oxidized and the sulfur remains open to form a disulfide bond. That's correct. Oxidation. Which class of proteins is the heat shock protein part of? Catalytic proteins, chaperone-like proteins, immunoglobulins, ribonucleolytic proteins. Yeah, I discussed about it previously as well. Chaperones are heat shock proteins of different classes. Class 1 has the heat shock proteins HSP70, class 2 has HSP60, that are the chaperonins. So yes, option B is correct. Okay. So there is a structure of a protein that is given. What type of structure is this? Is it a secondary structure, tertiary structure, quaternary structure, or a primary structure? So what type of structure is basically this? Okay. So what does the primary structure tell you of? The primary structure gives you just the amino acid sequence. If it would have been a secondary structure, there would have been only a few folds. There would have been just a few folds, maybe some alpha beta alpha or an alpha loop alpha, something like that. But a tertiary structure you see over here, it's a very well folded protein with secondary structures, some loops, alpha helices, so many things. And why is it not a quaternary structure? Because it's only in the form of a monomer. So tertiary structure is the correct one. In the hydrophobic core of a protein, which of the following forces is responsible for hydrophobic collapse? So for a hydrophobic collapse, which of the following force is responsible? Keeson force, Debye force, ionic forces, van der Waals forces. Okay, I get one answer. Anything else? So this hydrophobic, so when the hydrophobic interaction basically occurs, this is just a mere interaction, so that is mostly controlled by your van der Waals forces. Okay, van der Waals forces. So when like the water molecules are removed from the center, the hydrophobic residues occupy it, and the mostly stabilized by the van der Waals forces. So option D is the correct one over here. Yeah, that's a last question. That was. Anything else? Any other doubts? Anything else? Because I'm done. No, nothing. Okay, go through this. Anyways, I will be sharing the slides and the video will be uploaded in YouTube. You can check it. And if you have any doubts, you can ask me in the next class as well. Not a problem. Okay, then. Thank you for joining. Again, we will meet in the next week, Friday. Okay, thank you.